Basecamp is actually a pre-screening trial for a really interesting technology, which are logic-gated CAR T cells. And so this is really the first, hopefully, of a number of different trials. And one of the problems with CAR T cells, which have been very effective in hematologic malignancies, in solid tumors has been lack of efficacy, but in particular, on-target, off-tumor toxicity. So Basecamp is really sort of the feeder trial to a whole set of trials using a new technology with these logic-gated CAR T cells. What this involves is a target, which in this case is CEA, which is common in many GI cancers and other cancers as well. In addition, it has a blocker. So therefore, there's something that's missing in the tumor. And what's missing in the tumor is called loss of heterozygosity of HLA. So HLA-A2 is the most common HLA, at least in North America. We're using that to start out, but you could change that depending on what part of the world and what you want to do with this. And so it's relatively common, and it turns out that roughly about 20% of patients in their tumor early on have loss of that HLA. So the CAR T cells, which are in the follow-up trials, which will be called Everest, which should be starting relatively soon, they're engineered to have a target, which in this case is CEA, but we've already released data about mesothelin, and there are others that can be, so it doesn't have to be CEA. And then if you're HLA-A2 and your tumor has lost HLA-A2, therefore the CAR T cells would preferentially kill the tumor cells and leave the normal cells alone. And what Basecamp is a feeder trial to identify those patients and also to say, in real life, what is the incidence of this loss of A2, loss of heterozygosity. And in large data sets, it's been anywhere from like 15 to 30%. And what we showed was basically that's true. And that anywhere roughly around 20% of major GI malignancies would be eligible for this next series of trials. So we're very excited about that.